I know this is the last presentation of the day, but I hope you will enjoy listening these exciting results, at least the preliminary exciting results that we obtain together with Mons Nicolais and collaborators in Denmark on the composition of the microbiome in the Grapevine Yellows phytoplasma infected host. The purpose of this study was to explore the next generation's based method for the phytoplasma genetic diversity in single host, either plants or insects, and exploring phytoplasma interaction with the microbiome. And the main challenges of this study was to avoid the co-purification and possibly also the co-amplification of DNA of plant organelle, and to amplify selectively all bacteria, including phytoplasma. Generally speaking, the infected grapevine plant is like a microworld with a lot of bacteria that are living together and interact each other, and possibly interact also with the physiology of the plants. So the idea was to explore this microbiome using next generation sequences analysis. And we start from the samples, grapevine from Italy from 2019, and grapevine from Italy and Chile from 2021. And Mons, together with the collaborators, design a specific degenerate primer to amplify all the bacteria, including phytoplasmas, but avoiding the amplification of the plant DNA. And these primers actually amplify V5, V7 region of the 16S ribosome gene of almost all the bacteria. And the technical procedure for the generation of the samples. The samples were amplified with these primers, and they were purified from the agar gel to build the libraries for the next Illumina analysis. And these are the results that actually we obtained from all the samples analyzed in 2021. Together with the grapevine, here you see also the relative abundance of the different bacterial families in the different samples. So in citrus, palm, and insects, but they will talk about in the next days. So looking only on the grapevine samples, you see that the phytoplasma's abundance is very high compared to the other hosts. But looking deeply in the bacterial composition of the different grapevine from Chile and Italy, we found very huge differences. While almost 50% of the bacterial composition in the grapevine, Italian grapevine, was related to phytoplasma presence, in the Chilean grapevine, there were identified different bacterial families, and different from the ones identified also in Italy. And these were actually in agreement with the results that we obtained also from the normal PCR amplification in the lab, where very few samples from Chile were positive for phytoplasma presence. The samples used for this analysis were collected in Italy from 5 different regions, from the north to the south, and especially from Veneto, Emilia-Romagna, Tuscany, Abruzzo, and Sicily. And looking at the composition of the bacterial community in the grapevine from the different regions, you can feel also the big difference among the different geographic areas. Because while the grapevine from Veneto were the samples with the highest amount of phytoplasmas in the single plants, the almost 80% of the bacteria identified were actually phytoplasmas. In Emilia-Romagna, the percentage was a little bit lower, like 50%. In Abruzzo, even lower. In the last, in the rest of the regions, the phytoplasma presence was quite low and sometimes almost undetectable. And here in the graph, you can see the presence of phytoplasmas in individual samples. And again, the region where the phytoplasmas were easily detected was the Veneto, compared to the other region where only few samples were identified as phytoplasmas infected. And looking at the general beta diversity of the bacterial communities detected in the different grapevine from the different regions, here you can see the bacteria are forming clusters related to both the geographic origins and the grapevine varieties. So the composition of the bacterial communities is not the same according to these two variables. And these were the results of very preliminary analysis on the association and the correlation between phytoplasmas and the other bacteria. And you can see is a very complicated network of relationship between all these, among all these bacteria, and is not easy to understandable. And because actually the most informative, the most informative relationship would be the negative correlation actually of phytoplasmas with other bacteria. But these analyses are still in progress, so we don't know exactly which genera are negatively correlated with phytoplasmas. But as I said before, we start in 2019 with the preliminary analysis of samples collected in Italy. And I can show here better results that we obtained at that time, because we already analyzed, so I can tell you more about these samples. Actually, they were collected only from one region, from Tuscany, from two different locations in two subsequent years, 2018 and 2019, and from grapevine and alternative host, mainly bindweed. And here are the results on the bacterial composition in the different samples, and the phytoplasmas are the colored in pink. So as you can see from the graph, most of the grapevine samples were severely infected with phytoplasmas that actually are the most abundant bacteria inside the plants, while only few samples show a different composition in bacterial communities. And about the alternative host, only one sample of bindweed was only one sample was really was really affected by phytoplasmas. All the rest, the phytoplasmas were almost undetectable or very low percentage. But looking better inside the bacterial community and the bacteria families that we identify in the grapevines in the 2 years of sampling, almost 95% of the bacterial communities was composed by phytoplasmas, and only a small percentage was about other bacterial families. And also in the bindweed, only from one sample we could identify the coloplasmatase organism, so the phytoplasmas, together with other bacteria in less quantity. But looking to the case of one vineyard where the percentage of phytoplasma identified was lower, and also the symptoms observed in the vineyard were very low, we want to have a deeper look in the bacterial composition. And we found that the majority of the bacteria identified in this vineyard were actually belonging to the Enterobacteriaceae family, and mainly to two main genera, Panthoea and Enterobacter. So these two genera show a negative correlation with the phytoplasma presence in the plant. So at the end, what we can conclude from these preliminary results and from this preliminary data, that actually a next generation based method is useful to investigate the phytoplasma interaction with all the bacterial communities. And the study of positive and negative correlation among genera could actually help in understanding the pathogenicity of this microorganism. And in particular, the study of selected negatively correlated bacteria could eventually lead to the improvement of phytoplasma-associated disease control in the field. And finally, at the end, I would really like to thank Nicola Fiore from Chile for providing the samples, Mons Nicolaisen from Aarhus University, and the people at Aarhus University and University of Bologna that help us for processing and the samples for the bioinformatic analysis and for nucleic acid extraction. And again, thank all of you for your attention. Thank you.